we'll maybe move on in the interest of time. So the last talk is from Tim Wang, who's at the University of Sydney. And he's going to give us a sort of a discussion of a real-world clinical validation. So I think it's a nice sort of conclusion to the session. So focused on multiple sclerosis. So I'll let you take it away.
Thank you. Thanks. Before my talk and we'll load up the slides, can I ask how many of us have ever thought about the method you developed or the model you developed that can be implemented in the clinics to helping doctors, right? When you start the project, right? Many of us. So why we haven't done so? Why we haven't done so? So probably there is a lot of things, the challenges you never thought about it, or it can be very challenging, or potentially can be easier than you thought. So today's talk, I'm gonna walk through our journey essentially to put our ideas, to conceptualize the ideas to want to put machine learning model, helping doctors all the way to commercialization and all the journey and the learnings that we have. So hopefully give us a little bit of feeling about how we translate actually one of the methods into real world clinical practice and validate that. Right, so just quick my disclosure. By the way, I'm an electronic engineering background, so signal and imaging processing, this has led me into the field that I'm in today. So, as we know, many of neurologic disease progress not necessarily have the symptom. For example, in multiple sclerosis, right? So that is actually today a lot of the therapies is they're not only targeting to stop the clinical relapses, to stop the clinical progression, they're also looking for stop the MRI progression. The evidence that we can find from our MRI, there is just no brain deterioration because of the disease. So that is why we're doing a lot of biomarker research, we correlate with the clinical findings, and we always have those problems. So for example, I'm just quickly show you, this is essentially, it's very obvious that this is progression in multiple sclerosis over time. You see the lesions expanding in the brain, but many times is we're really looking for something really subtle in the brain that we are sometimes really hard to see even using your naked eyes. So to build a reliable quantitative imaging biomarker to use at individual level, so we have a lot of the challenges we have to face. For example, this is a slide I'm just showing when I was a neuroimaging analyst, how I perform lesion analysis back in like 10 years ago to precisely quantifying the lesion change over time. I have to go through slice by slice and the building the evidence in the clinical trials as to demonstrate the treatment efficacy. If we wanted to implement this type of technology in individual clinics, this is the common three problems that we have to facing. We have a biological and a technical variability. This is we know when we develop a model working in one data set, it's not gonna work on the other data set, right? Second, it's a really complex analysis process from all the way of the imaging acquired at MRI scanner to be interpreted by the radiologist or by the neurologist. And the third challenge is really about, okay, if I can tell you the patient's lesion volume is 4.8 milliliter, what that really means to my patient's disease progression. So those are the challenges that we have to address to demonstrate the efficacy or demonstrate the gains that can help doctors in the clinical practice. So as we know, so essentially we had a great opportunity back in the days 7 years ago, and so we partnered with NVIDIA, fortunately got a CRCP to put this collaboration together, really to pushing the artificial intelligence in helping us to improve the biomarker detection at the clinics. So as you know, this is probably a very old AI technology. As you know, everyone is focusing on foundation model now. Back in the day, we're still doing a lot of the units, and that just actually works very super well for us in a lot of those disease activity identification. So yes, as you know, published papers, and this is probably a lot of researcher does as well, publish papers that stop there and moving on and other better methods. So I think the next couple of slides is really what I wanted to focusing on is the journey that bring this type of technology into clinical practice. So when you have like amazing amazing model, right? So you generate dice overlapping coefficients like a 0.9 or 0.8, something superior, the state of art. But when you implement it, you always found your methods is much more than just a push a button to generate the results. Because the first challenge you're facing is the variances in the MRI analysis. The variances that come biologically and also from the technically as well. So, we have all this problem of, different MRI fields, the MRI windows, the models, the coils, the software versions, the sequences. All of those things will bring additional challenges to you. Surprisingly, I should say not surprisingly, this is actually the most time and effort in our team that we spent to tackle those problems. Because before we're doing this project, we're actually running central imaging analysis services for pharmaceutical company for phase 2 to phase 4 clinical trials. We received all different type of MRI protocols in our center. So we can see the variances that are generated. Some of them are obvious, some of them are not really obvious. I just to give you one example, you could really appreciate that. So for example, this is one of the cases when we're doing the brain volume analysis. I received from this center a pair scans. I think many of us already run the Sienna analysis looking at the brain longitudinal brain volume change, right? So when I doing the analysis for this specific data set, I noticed this 10 patients has consistently demonstrated brain shrinking, right? This is multiple sclerosis patients, right? So you can see the percentage brain volume shrinking. Some of them is over 1%. But from what I know, our doctors there, doctors treating our patients really well. It's just very surprisingly see this is very high brain shrinking in this data set. I don't know why, and then I went through at length trying to understand what's really happening. I put the imaging side by side. I couldn't tell the differences. It's all from one single scanner, consistent coil acquisition. Everything is controlled. And when we look in deeply, I put them together. You'll realize this is flipping. This is actually from one patient just about a couple of months away. The difference is because one of the distortion correction function did not turn it on in the follow-up, right? This is one very, very easy to be missed, especially if your scans is acquiring at large hospitals, at very busy clinical environments, and collected it from the real life. This is type of scenario. Sometimes the radiographers. It's definitely not, I won't say that it's a mistake or something. It's because for the purpose of the diagnosis, you not necessarily need this level of the precision and precision to acquiring the scans. So this is actually the main cause generated the variances in the brain imaging analysis. You think about it, for those challenges and the problems, this is what we needed to identify, correcting, in our final analysis. So there are many, many examples of that. So this is just to give you a little bit flavor. That's the time we spent on this. So eventually, fortunately, we put all the models, a lot of imaging analysis in terms of the quantifying the brain structure, quantifying lesion activity change into this one single report to be able to fully automatically identify disease activity between the time points for patients with multiple sclerosis. So as you can see, the red one just indicates chronic lesions, no change from last time points, and the green one indicates slow progression. And what I'm showing you on the top right is one patient, another patient. So as you can see, the concept is actually not complicated. It's just very simple. Essentially just helping the radiologist to identify those very subtle changes in their follow-up time period to see if there is necessary to escalate the treatment, to change the treatment, or just simply understanding the disease progression. And actually, a lot of those time, and the patients not necessarily presented any symptom clinically. So this is so far using a lot of units, a lot of simple machine learning methods to generate those masks. Second biggest challenge. So when you have amazing robust model, so only probably 50% success. because the other 50% of success really in how you're presenting data to the doctors. The doctors do work in a very busy environment. I still remember our radiologist, consulting radiologist told us, if you ask me to open another software to click one button to see your results, this is probably take me to thinking twice to use your algorithms, because they have to put through under a lot of pressure to put through the numbers. So this is we actually need to design something really embedded in their clinical workflow. So this is essentially for radiologists, right? So what we did, instead of going ahead to develop a new interface, new software, we coming back to the DICOMs. You will realize how much powerful the DICOM protocol are. You can embed a lot of the methods, a lot of the displaying of your results, even PDF reports into the DICOM. As long as the DICOM can embed those information, it can be read by all the existing PACS system, right? So you automatically get the integration with the clinical workflow. So when the radiologist see your analysis results, they just looks like just seeing another sequences acquired at MRI. So they can interpret at the time when they see this case. So this is really where we found the niche that can embedding those AI models, AI algorithms into the clinical workflow. So as you know, as the part of submitting to TGA FDA approval, we have to demonstrate the efficacy, the utility of our software and the benefit to the clinics. And this is actually as the part of evidence we submit to FDA and the TGA. So it's basically one of the largest real-world clinical validation for this AI-based MRI monitoring for multiple sclerosis. We did about almost 400 patient pairs for 400 pairs of MRI from 282 MS patients. This is all real-life data. We did a little bit trick here. Essentially, we didn't do a prospective study. This is all done under retrospective phases. So this is actually tremendous of the saving the resources to do so. So what we did is we comparing the AI models, radiologist reports, and also the central imaging services analysis for disease activity, to looking at the new enlarging and the brain atrophy to identify if there is any agreement or the improvement. Not surprisingly, so using the machine learning model, of course, we could almost double the sensitivity from the radiologist report, how they're picking up the disease activity. Because as you could see from the previous example, because some of those pathologies are really subtle, so especially when patients with a lot of pathology in the brain, so using the deep learning, of course, that we can identify much more disease activity to assist in the clinical decision making. So this is absolutely not at losing the specificity. We also compared the brain atrophy, because we know the accelerated brain volume loss is actually getting more and more attention these days, than potentially now, not only just in multiple sclerosis, but in all the other diseases as well. So as a normal aging, we have 0.1 to 0.3% brain volume loss per year. Because of pathology, it's potentially much faster. And that is actually not necessarily presenting the symptoms. So this is another biomarker more than just lesion activity. So when we compare them, we realize the brain atrophy has almost never reported in the radiologist report, because it's just simply impossible to be identified by the radiologist from their reports, because this is subtle. You think about it, you're looking at a baseline of follow-up imaging. If you wanted to look at a 0.8% brain volume change, which is almost double the normal aging acceleration process, this is almost impossible. So this is another potential good use of this type of models in the clinical decision making. Another evidence that I want to quickly show you is there is a lot of incidentals in this report. So this is another thing for the old UNet model. We're not necessarily able to address all the problems that we have in the BC clinic. So we need a much powerful model these days that can coping with not just the lesion identification, but identify many other neurological, many other incidentals or the disease, the patterns there. So regarding interpretation of those results, how we make sense of the individual measurements of those lesion volume and the brain volume change, right? So first of all, we've done a very quick comparison just about how doctors are classifying the disease loads in the brain versus what is the actual disease load in terms of lesions in the brain. So doctors qualitatively describe the words: severe, single lesion, a little bit a moderate, heavy load, or very severe lesion loads. And quantitatively, we quantify all the scans into different centile, right? So we found we actually can match almost 2/3 of the subject with the doctor's description, describe of what is the disease load. But the difference is really on this 1/3. Do you think, can you guess what is the doctor's normal patterns? Are they usually likely to underestimate the disease load, or do you think they are overestimate the disease load? They actually tend to describe underestimate. So they usually describe a moderate more often. Actually, the disease load is more towards to the last, the higher centiles of the disease activity. So given the time a little bit over, so essentially just to quickly cover this one as well, how we put the interpretation of this 4.8 milliliter T2 lesion volume. I think this is potentially can be useful if you done a large cohort study, you've done some predictive power for your patients' predictive model, how you make that sense to individual patients. So this is what we have done. So what we did is we grouped, this is the design, we group all the patients with the similar disease load in terms of, for example, in this case, is lesion volume, right? So you think about this patient is 4.8 milliliter. In my database, large database, I categorized all the patients with 4.8 milliliter. Then I start comparing with the similar disease duration. What is the age distribution of this specific patient? In this patients group have a very similar disease load, right? For example, in these patients, it's actually sitting at a very senior age. So this is actually a little bit hint of to the physicians. Okay, even with the diseased cohort, these patients actually at a very senior age. Comparing if these patients at a very young age, there may be something you need to think about it to actually to give your treatment for that. So at the last, I want to just quickly, also this is another implementation for research community. This type of the software already made available to MS-based imaging repository completely at no cost. So every single imaging analysis that the research community contributed to the MS-based imaging repository will have this type of analysis. This is another way how we implemented that for the research community. And of course, the clinically and the models that were already implemented in many of the hospitals in Australia and in the US. Thank you. I think that's pretty much all. And thanks all, forgot to mention, acknowledge for my amazing group here and also the collaborators over the years. Yeah, thank you.
Any burning question for Dr. Wang before we move on to a more general question and answer session?
Yes, I have a question about the lesion load measure. I'm a little bit surprised sometimes in MS you haven't yet established kind of a weighting map how important it is where the lesion in the brain is. What's the state of the art there?
Yes, and this is another stream of my research actually, and you're absolutely right. The location is just like a real estate. The location matters a lot. It's absolutely more, and it's definitely directly linked to the symptom, to that. And we haven't really, we have something research, but haven't really implemented in this type of report yet. Yeah, but you're absolutely right. For example, lesion at the brainstem and the internal capsules is much more important than in the deep white matter. Yeah.
Do the clinicians use the location of the lesion, or are they more about just the volume?
So, from my understanding, a non-clinician, they often for the treatments they're looking at the changing over time, because a lot of the treatment is really to stop the MRI evidence to demonstrate there's new lesions, right? So, if you have even one to couple of new lesions that appear in the brain, that means that probably something doesn't work well, and they need to either escalate the treatment or not. But in the initial reporting for the best, I think probably for the patient's first consultation, they're probably looking at the location. But this is from non-clinician. There may be.
What I was thinking was, you mentioned some practical barriers. You probably, the clinicians need to be consulted about how they use location before you even bother building a technology that works for finding the location, right? Yeah.
So, Nicholas Port, Indiana University. I'm a neuro-ophthalmologist. So, in the vision world, we spend a lot of time worrying about location, and particularly we're also concerned about is not in the optic nerve when you have hyperintensity flares. The optic nerve degenerates as a result of that, and so we are monitoring, particularly tracking optic nerve flares, and then using OCT to measure how much of the retina has died, because as we call it the island of vision solely sync. So yeah, locations really matter, especially when you're a vision geek like me. So.
Yes, absolutely. And the vision system is actually my favorite system that I use to monitoring disease progression. We do actually have a composite electrophysiology and imaging biomarker using vision system as the window to monitoring the disease progression in multiple sclerosis for pharma. Yeah, for trials. Absolutely right.